So we have picking out a new puppy here. The quote from Gregor Mendel is, "My scientific studies have afforded me great gratification. I'm convinced that it will not be long before the whole world acknowledges the results of my work." A little bold, but when you're right, you're right. So our learning goals are to understand the influence of Gregor Mendel's experiments, describe inheritance using the proper vocabulary, distinguish between dominant and recessive, homozygous and heterozygous. So we've already talked about DNA in terms of a biomolecule that encodes all the information in our cells, how DNA is packaged up as chromosomes, and that chromosomes are what's transferred in the processes of mitosis, meiosis, and fertilization. That's what's carrying all of our genetic material, and that's where traits are going from one to another. So whenever we're looking at the lab, we were looking at our puppies, we realized that some traits are passed down from parent to child, but not all. And seeing a parent's traits can be a reasonable but imperfect way to predict a child's traits. Some traits are more likely to inherit than others. Whenever you look at enough examples, you see different degrees of consistency. Some things show up a lot, some things show up sometimes, but not others. Some things almost never show up. But, just looking at them physically doesn't give us a very good level of prediction of what will happen in the young. So, in the early 1800s, Gregor Mendel became a friar because at that point in time, education wasn't widely available to everyone. But friars were places where learning could take place. So, he ended up with this title, the father of modern genetics, because during his tenure, he spent a lot of time not just doing experiments, but keeping data and analyzing that data. It took a long time, but he became famous posthumously, meaning after he died, when his work was later recognized in the 1900s. So genetics is the name of this branch of biology. Biology is a large subject that has lots of small subjects in it, and one of those branches is genetics. And it's all about heredity. Heredity meaning how we get the traits that we have and how they come from our parents and the people that come before us. When we're talking about traits that we're inheriting, those are just all the different characteristics. Our height, our muscular structure, the color of our eyes, how our organs do what they do, all of those things. So data collection was a really important part about this because we needed to have organized information that we could look at over time so that we could draw the conclusions from it. So first, Mendel recognizes that there's a diversity among the plants, meaning that whenever you look at a large population of them, they don't all look the same. There's variation. Some of them have different kinds of seed shape, seed color. The seed coats are different. The shape of the pod, the color of the pod, the way the flowers are growing on the plant, whether they're short or long. All these different things made each different pea plant unique. So there obviously were more than just these 7, but these were the ones that he's collected the data about and studied. So when he goes and looks through all the plants that are growing, and he looks at one set of plants to another set of plants, he started to notice some patterns. This is the beginning of the need for experimentation. So he was looking at one garden that had only green pea pods. And when they produced seeds, and he grew new peas from the seeds from the green pea pods, they always grew new green pods. And then another one where there were yellow pea pods. And in that garden, when he took seeds from the yellow pea pods, they produced seeds that always grew plants with yellow pea pods. That was pretty normal. But what was weird was he took one of the green plants and one of the yellow plants and let them breed together. The pollen from one mixed the flower in another, and he got seeds that were produced from those two plants. But all of the seeds that came out, even though it came from a green and a yellow breeding together, they always come out green, and there was never a yellow. So the green pod was true breeding. True breeding means that it always bred out green. The yellow pod was true breeding, meaning it always bred out yellow. But together, the F1 generation, the daughter generation, would get all green. Now, if you took two of those green pods that grew from the green parent and yellow parent, two of the pods from the F1 generation, and you bred those together, it was pretty consistent that for every 3 green plants, you would get 1 yellow plant. We could also say it's 75% green, 25% yellow. And, if you grew a whole bunch of plants like that and you went counted up, it was this ratio, this 3-to-1 ratio that kept coming out again and again. And whenever he looked at all 7 traits, he noticed a pattern like that as well. If you look there in the data table, this is one of the data tables from that Mendel collected. You can look at some of the numbers are very large. For example, let's just look at that first row, the form of the seed. He crossed one that was a plant that had round seeds versus a plant that had wrinkled seeds. And in the first generation, they all came out round. But then when he crossed round with round from the F1 generation, in the F2 generation, we got some round, some wrinkled. And look how many plants he studied. 5,474 of them came out round, and 1,850 of them came out wrinkled. Having that large of a sample size of information really gives us a lot of confidence in his results. And you can see 5,474 is about 3 times as large as 1,850. That's that 3-to-1 ratio. This tells us that he was onto something, that this pattern is coming up. So we need to define a couple words here: gene and allele. So we all have different genes. A gene is just part of the DNA, because we know that DNA codes for all of our traits. So if we look at how much DNA you need to describe one trait, like the shape of an earlobe, that section of DNA, whether it's a couple hundred bases or a couple thousand or a couple million, however long that string of bases is, the string of bases that codes for one gene is called, for one trait is a gene. Now, alleles are different types of that gene. So for the gene that codes for earlobes, there'd be one allele that's an attached, and there's one allele that's for detached. So some of us have different alleles for different ones, and that's what determines our trait, what allele we have for that segment of that gene. So what Mendel concluded is that there was more than just one allele that was determining that traits. That we're not just this or just that, but that there were 2 alleles joined together in every organism for every trait. And both alleles together determine how the trait comes out. So when those 2 alleles are together, some alleles end up being dominant over others. The dominant alleles always express their trait. So if you have a dominant allele and a recessive allele, then we're going to get, whenever you look at the physical form, you'll see what the dominant allele codes for. If you have 2 dominant alleles of the same, obviously we'll see that one. And if you have 2 recessive alleles, there's no dominant allele around to dominate them. The only time we'll see the recessive allele is if we only have those 2. Here's a developing question for you. So we might talk about here how in their DNA, the DNA codes for all of their different characteristics. And whenever we look at them, you can describe some of their traits, whether they had the trait for this kind of pattern or coloring, or the trait for this ear shape, or the trait for the size of their snout. And then we could talk about what alleles the parents might have had, whether they're dominant or recessive, and how that comes out in the child. And that segment of the DNA, that gene, is what we're looking at. So in the experiment, which allele that coded for the pea pod color gene was dominant, which allele was recessive? I'll let you pause here and think about this. So if when we took the true breeding greens, you across the green garden always got green plants, and you took the yellow plant from the garden that always got yellow plants. When you crossed them together, all of the F1 generation came out green. That means that F1 generation got one allele from the green plant and one allele from the yellow. Well, when you have one green and one yellow allele, but your trait ends up looking like green, the green is dominating over the recessive yellow. So we have some shorthand to write like this. You know, we don't want to always write everything out in long sentences. So whenever we write, we give the dominant alleles a capital letter and the recessive alleles a lowercase letter. So those green pea pods the ones that were true breeding from the first green garden that always got green plants, they must have had both capital G's, both dominant traits. We know that because they're true breeding, that means there are no recessive ones anywhere, because yellow never showed up in the true breeding green garden. In the yellow garden that was true breeding, they must have had two recessive traits, because if there is even one green allele in there, they would have showed up green. So the fact that they were yellow, we knew that they had both lowercase g's. So whenever we describe which alleles you have, we call that your genotype. So the genotype of the green pea from the green garden was capital G, capital G. The phenotype refers to how the trait comes out to that. So the phenotype was the green color. Now, the genotype and the phenotype can be very similar, but they tell us different information. Genotype is what is the code, and phenotype is what did that produce. So here's a proficient level question. Think about what is the phenotype of this pea pod, what is the genotype. Think about the difference in those phrases. I'll let you pause and think. Now it's phenotype here. Phenotype is what it looks like. So the phenotype is, that's a yellow pod. Now, there's only one kind of genotype that codes for that. It must be two lowercase g's, two recessive alleles for the color. Because it could either be capital G, or it could be a capital G, lowercase g, or it could be a lowercase g. Those are only three combinations. The first two have green phenotypes, though. Only lowercase g will get that yellow pod. And that's the difference. Here's a mastery level question for you to think about. What led Mendel to believe that there were only two alleles instead of one or three? Because he didn't have advanced microscopes. He couldn't look down and see the chromosomes or look at the DNA and count them. We've been able to confirm this now with the advanced technology we have in our modern era with electron microscopes and, those kinds of really advanced technology. But he didn't have that. What evidence did he have to get two? I'll let you pause and think. Okay, the evidence, it was in those ratios. That 3-to-1 ratio whenever he bred the F2 generation, that comes from 2 times 2. If there would be only one allele, then you wouldn't get any of those ratios at all. You'd always get the same as the parent. And if there were 3, well, then you get an even wider variety of different combinations coming out. You get a much more complex ratio. That 3-to-1 ratio, that shows us 4 options, which it comes from 2 squared. So homozygous is the phrase we use whenever your alleles match. You're either capital G, or your lowercase g, meaning that if two homozygous parents breed and they have the same trait, you're going to see the same thing in the young. The opposite of this would be heterozygous. Heterozygous means that you could get either one. So it'd be a capital G and a lowercase g. So the F1 generation in Mendel's experiment was heterozygous. So if we look at the homozygous green from this plant, if you bred it with a heterozygous one that had capital G, lowercase g, well, then you could get either green or yellow. Heterozygous parents can have different kinds of kids. So if we look at the genotypes here in this cross, if we took the true breeding green, the true breeding yellow, capital G, and lowercase g, you can see that they all come out green. They're all heterozygous in this case. But because green is dominant, even though they have the heterozygous genotype, they all have the same phenotype of green. If you look at the bottom here, whenever we took seeds from the F1 generation and we crossed to the two heterozygous green plants, then we got that 3-to-1 ratio of green to yellow. One of them came out homozygous green, two of them came out heterozygous green, and one of them came out homozygous yellow. But even though there's 3 different genotypes, there's only 2 different phenotypes. So we see 3 green and 1 yellow. So that's all the information we have on how Mendel's experiments work. It's important to note the different vocabulary phrases and see the logic of how Mendel delved into this information, just given the data he had from looking at gardens.